In this video, we will discuss about this lipo-nanoplatform that they have optimized for the in vivo delivery. In the previous video, we have understood the efficacies of their lipo-nanoplatform, but that nanoplatform was not optimized for the in vivo delivery. So in order to optimize for in vivo delivery, what they did is they modified their bilipid layer by adding these three components, that is dope, DSPE-PEG and CHEMS, and together they form a liposome, and this will help this they have used for the formation of this nanocomplex. And the more information about this nanocomplex, you can refer to the previous video where we have discussed about the compositions of the nanocomplex. That is, it contains this OPSA, which is ROS reactive. So continuing our discussions, so thereafter, once they form this modified lipo nanocomplex, they characterize this lipo nanocomplex using cryo-TEM, and they are able to find out that the diameter is around 110 nanometers. This and the cryo-TEM images are shown in this panel C, and the average diameter is in the panel B. And once they characterized it in terms of their hydrodynamic diameters, then they used it to compare it with the traditionally transfected approach, that is PEI. So once on one side they use the PEI lipo-nanocomplex, and on other side they use OPSA based lipo-nanocomplex, and they can see that OPSA based complex functioning well, and the functions that they are checking is that whether it is able to make pores or not. And pores are important so that they can use those pores for the internalizations of these granzymes, which is essential for the activation of GSDMB. And that part, how this GSDMB and granzymes are connected, we have also covered that part in our previous video. So in this case, we can see that the pore size is high, and also the number of pores are high in their optimized conditions, that is OPSA with lipo nanocomplex. And these things they have quantified in the panel E and F, where the panel E shows the number of holes, which is significantly higher, and also the diameter of holes, which is higher in their optimized conditions compared to the traditionally transfected conditions, traditionally transfected approach using PEI. And thereafter they wanted to check whether this pore size is able to do functions for which they have developed this pore size, that is whether it is able to transfect some big size molecules or not. So for that they use these FITC molecules, which is practically impermeable for the to transfect on its own. So for the control, they do not use any transfections, and with this traditionally PEI based lipo-nanocomplex, they do not see any FITC signals. But with this OPSA lipo-nanocomplex, they are able to see the FITC signals, indicating that the pore size is big enough to even transfect, even make this FITC permeabilized inside the cells. And these things they are quantified using the flow cytometer, where the representative image shows a high intensities of this OPSA lipo-nanocomplex, and its quantified image shows a high fluorescence intensities compared to the controls and their traditionally used approach PEI. So this shows the reproducibility is also quite high. However, one thing that come into the researcher's mind is that when this pore size is high, then the membrane might lose its fluidity, or it might be weakened. So in order to check whether the membrane is weakened or not, they use this FRAP based approach, where they check if the intensities goes down, indicating that membrane's fluidity is weak, and or the in normal case, or in simple terms, the membrane is weakened if the intensity goes down. And they can see that in the pre-bleach conditions, the fluorescence of the membrane seems to be intact, and after the bleach conditions, that is during the transfections of this OPSA based lipo-nanocomplex, the intensity goes down, and after some times it is recovered, indicating that the membrane is again recovered after some times. And these things they have also quantified using the normalized fluorescence intensity. They can see that after like 50 minutes or 1 hour, after that around 50 minutes, they can see a decrease in the pore size or decrease in the fluorescence intensity, indicating the membrane is more fluid or weakened, or the pores are there, because of that there is no fluorescence available in that region, and after some times they are able to see a recovery. So they can see the recovery. So this indicates that there is a temporary weakening of the membranes of these T41 cancer cells that they have used. So after that, they, once validating that their system is so far functioning as per the expectations, so now they have one more idea, that whether they can use this time period of this 1 hour to also internalize this granzyme, so that it can make this GSDBM, it can do the proteolytic cleavage of GSDBM and make them into its active form. So for that, they compare two different approach. First, their idea that whether they can use those time period using this, whether they can use that time period to also internalize this granzyme, and compare it with the traditionally electroporation approach. And so what they did is, they took the cells and transferred them with lipo-nanocomplex and the granzymes together, and in other case they electroporate them and then electroporate this FITC based FITC and this granzymes, and then check the signals, and they are able to see that both the conditions, electroporations and OPSA-lipoNanocomplex, shows the same fluorescence. And these things, fluorescence means this FITC signals, and these things they have quantified using mean fluorescence intensity, where the signals are also similar, and in terms of this positive rate, that is number of cells that gets transfected, is also similar. And when they check the cell viabilities, then cell viabilities are also, they are slightly high in the case of this OPSA based lipo-nanocomplex compared to the electroporations. And once they find out that their system is able to work properly, the final diagram that they have used in the final idea that they have when they co-culture both granzymes and lipo-nanocomplex together with cancer cells, is that the lipo-nanocomplex will internalize through the membrane fusion, and at the same time these granzymes will also be able to internalize or perforate inside the cells, and thereafter this lipo-nanocomplex along with this lipo-nanocomplex will be able to form, or lipo-nanocomplex will be able to form, that is convert this OPSA into its active form PSA, and at the same time these GSDBM formations will also occur, that will also be transduced in this lipo-nanocomplex. And this GSDBM which are formed, that will be cleaved by these granzymes, and that will cause the pyroptosis. So first they check whether the activations of this GSDBM occurs or not, and they can verify that GSDBM activated GSDBM are formed in the case where they co-culture both lipo-nanocomplex and pGSDBM along with the granzyme, but not in other cases. And so once they confirm that GSDBM active form is present within the cells, tumor cells, then they check whether the perforations are occurred or not, and they are able to see that the perforated or this pyroptosis occurs or not. So they are able to see that pyroptosis signals using the using this microscope images. You can clearly see the pyroptosis of the cell that is marked by yellow color, and the whole process that is going on within this is shown in the panel S. Here we can see that once this GSDBM is formed and these granzymes are present because of this co-culture of this lipid nanoparticles and granzymes, the GSDBM will be cleaved off to make it into the active form, that is GSDBM N, and represents the N terminus, and that will cause the pyroptosis that results in the release of the pro-inflammatory cytokines, and that will further activate the immune systems to act against the tumor cells. So this is how they checked and verify the lipo-nanocomplex method for the in vivo delivery. I hope you understood to some extent how this method works, and how they validate their lipo-nanoplatforms. If you need to understand more about these experiments and also wants to look the supplemental figures, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.